Thank you for the very kind introduction. Yes, it's my pleasure to be here to summarize my PhD research on accelerating liquid formulation design using lab automation machine learning. I've been supervised by Professors Alexei Lapkin and Kedar Hippogönko over in Singapore, and our work has been in collaboration with BSF Shanghai. As part of my research, I got to spend a couple of years abroad at Cambridge Cares and A-Star in Singapore, and my first and fourth and final year here in Cambridge. Oh, sorry. Okay, perfect. So firstly, what is a liquid formulation? Liquid formulations are composed of several ingredients which are selected, processed, and combined in a specific way to obtain well-defined functions. So we have on screen some example liquid formulated products from paints, detergents, cough syrups, some sunscreen, and my work focused on shampoo formulations. More broadly, the results and methods that are developed in my PhD are applicable to surfactant-based products in the personal care industry. At the start of the project, we were given the following problem statement from BSF, that an effective way to establish or predict the relationship between formulation ingredients and the desired formulated product properties is required. So particularly with the drive to develop more innovative products, to develop more sustainable products, or to comply with changing regulatory or supply chain pressures, being able to predict or rapidly screen formulations would be a big win. This is, however, easier said than done. Formulations are complex mixtures. If we think about shampoo, for example, we've got small molecules like the surfactants which are responsible for cleaning your hair. We've got macromolecular ingredients like the conditioning polymer in 2-in-1 shampoo conditioner products. And if we try to use first principles, models, predictions, approaches, these are going to be very difficult or almost intractable to get the formulation property predictions. And also we are not able to rely on physical chemistry knowledge alone to be able to make these predictions. We have, however, shown in an earlier study that we're able to use machine learning to learn over the complex interactions in these formulations to make relatively cheap and accurate property predictions. Now, as you all probably know, with any data-driven approach, the experimental data is the most critical factor, and unfortunately here, the availability and ability to generate experimental data is limited, and therefore I would say the largest focus of my PhD was in attempting to set up a high-throughput liquid formulations workflow over in Singapore. Now, in formulations development, you have traditionally lengthy and manual characterization or processing steps, so there were several hurdles to be overcome here. Now, once we're able to perform the experiments, we still have a very vast experimental design space. So our experimental budget is relatively limited in relation to the space we want to screen. Therefore, this leads to the question of how do we select which experiments to perform? And this was another chapter of my thesis. And once we're able to collect the data and we want to machine learn models over this data, we have the question of how to featureize these formulations. So here, when we're speaking about featurizing chemistry, what we mean is how do we encode the chemistry in a way that the computer can understand? Now, there are several established methods for doing this with small molecules and in pharma research and things like that. And during the course of my PhD, in parallel, others have been working on emerging methods and works for polymers and polymer property prediction, which is also a very exciting field. However, with formulations, we're looking at a complex mixture of these small molecules and macromolecular ingredients, which makes this a challenge. So this slide summarizes an overview of the semi-automated machine learning-driven workflow that I developed in Singapore. It starts with a design for experiments work list, which suggests which experiments to perform. These were then executed on the Opentron's OT2 liquid handling robot, which we retrofit with a mass balance to be able to do viscous liquids handling. The formulations were then carried over to a multi-position magnetic stirrer and mixed for a half hour, after which we assess the formulations for stability by a visual inspection by eye. We also set up, as you can maybe see, we set up an imaging station to collect images for each of the formulations, and I later showed that we can train a computer vision classifier to predict whether the formulations are stable or not. The stable formulations were then carried over to an in-house built pH bot for fully automated titration of the viscous liquid formulations. after which we waited a pre-agreed 36 hours before reassessing the formulations for their stability and carrying forward the stable products for the final characterization of turbidity, for which we measured UV-vis absorbance as a proxy measurement. And for the rheology measurements, we had to resort to offline rotational rheometry. So also through my research, I was looking into any sort of high throughput or automated methods for viscosity characterization, but this is still an open problem. And if anyone is interested in this in this audience, then I would love to have a chat about that afterwards. On this experimental workflow, we ran an 8-month experimental campaign and generated over 800 liquid formulations. And this data set and workflow has recently been accepted to scientific data, so hopefully this, along with all the ingredient structures, will be published soon. The workflow started with the design of experiments, and we're looking at preparing formulations with 2 surfactants, a conditioning polymer, and a thickener in a base of water. We could choose from 12 surfactants, 4 polymers, and 2 thickeners, resulting in 528 possible ingredient combinations. In addition to selecting which ingredients to use, we want to decide what concentrations to use them at. So we went back and forth with BASF, and I agreed upon a set of concentration ranges that we were going to explore. Overall, you have a very high dimensional design space, and this is a problem typical in formulation design. So now if we look at this figure, we have the first step of suggesting which experiments to perform. We're going to prepare the formulations and ultimately only obtain viscosity and turbidity data for the stable formulations. So the key point I would like you to remember here is that the unstable formulations result in partially wasted experiments. We went with the idea of having a space-filling design, and I'll now introduce some background on these space-filling designs, because the objective of this work, unlike the previous talk which was discussing Bayesian optimization, was to collect as informative a data set as we could to then be able to build the forward model for the property prediction. So with the space-filling design, where you're trying to be as representative and sample all of the design space, then when you develop models and machine learning predictions over these, the predictions should ideally be close to some other point that you sampled or was in your data set. So with space-filling designs, an intuitive criteria you can think about for how you would develop one is you could try to maximize the spread of the points or maximize the coverage of the points. And this is what is known as maximin and minimax designs in the literature. Typically, minimax designs are actually quite expensive to compute, so we often see maximin designs being used. However, one of the issues you can see with these 4 points in a simple 2-dimensional space is that they can have redundant projections. This is why we also commonly see in the field Latin hypercube designs being used. These have good space-filling properties in the full p-dimensional space, so p being the factors we're looking at, and they have uniform 1D projections. Now, with a Latin hypercube design, you can have different ways of having a Latin hypercube design, and typically when people are referring to they use Latin hypercube sampling, they actually mean they're using maximin Latin hypercube designs according to the Morrison-Mitchell criteria from 1996. Now, recently in the last decade, the group of Roche and Joseph at Georgia Tech came along with this new MaxPro method. A maximum projection design is a type of Latin hypercube design subject to the MaxPro criteria, which has better space-filling properties in all other subsets of factors. So where I'm looking at formulations with surfactants, polymers, thickeners, if I say fix the thickener and want to see how is the response of particular surfactants with this thickener, and I'm not looking in the full p dimensions, but here p-1, I fix something, then actually the MaxPro design is better. This group in Georgia Tech, they published a really nice package called MaxPro QQ, and the QQ here stands for quantitative and qualitative, and this was one of the key challenges in developing a new DOE method, that I'm looking at a mixed variable design problem. And then I took inspiration from this idea of having a weighted space-filling design, where weighting is meant to minimize sampling in regions of little relevance, and as I alluded to earlier, this was the unstable regions. So we tried to direct the experiments to predicted stable regions, and as we do not know a priori which these stable regions would be, we're training a stability classifier in an active learning cycle. And also in this work, we introduced a featurization by the surfactant functional groups. So we're trying to move towards having more generalizable property predictions. Moving back to the experimental side, the first experimental step was automated viscous liquid handling on this OpenTrans robot. I had the choice between two different liquid handling robots in the lab that I was in, and favored using the OpenTrans for its ease of use and flexibility in programming different liquid handling parameters. So I could try to optimize the liquid handling parameters for some of the highly viscous ingredients that I had, but ultimately we were still not able to accurately and reliably dispense the target amounts from our design of experiments. So we went to installing a mass balance underneath the OpenTrans, and I developed an automated gravimetric analysis protocol to then be able to reliably know what is the composition of the formulations that I am preparing. And in a separate side project, I also looked at leveraging the error from air displacement pipetting of highly viscous fluids to correlate this with the fluid's viscosity itself, and developed a high throughput proxy viscometer for Newtonian fluids, which was published in Digital Discovery. Now, a key step of the formulations workflow was pH adjustment to industry-specified target using citric acid NaOH. These are titrants. If you look at the back of any product at home, personal care product, you usually see the citric acid listed as an ingredient. This is because as a weak polyprotic acid, it's able to confer good buffering properties so that the formulation remains stable at the target pH for longer. With this work, we built a fully automated pH bot capable of lab-scale batch titrations of viscous formulations. And since we were using a weak polyprotic acid, this has in fact necessitated the development of a new method, a new titration algorithm. We had some previous work in the group led by Alexander, who's a recent graduate and now co-founder of ReactWise, who developed a machine learning-driven titration algorithm, but this could only work with strong acids or bases. So I had to go and extend the method, which I'll present on the next slide. Another challenge we had was that I'm working with highly viscous formulations, and so we had an engineering challenge in terms of designing the pH bot appropriately for full automation. We had to optimize the cleaning and mixing protocols, and we were able to do so. So over here, you can see I've developed a physics-informed machine learning-driven titration algorithm, where the idea is if we knew beforehand, when we're doing a pH adjustment, what the titration curve for that adjustment would be, then we would be able to do a simple one-shot approach. We would know exactly how much acid or base is required to reach the target pH. However, we don't know this beforehand, but we can train a surrogate model. Here you see on screen a Gaussian process surrogate model. And it's being trained over the net moles of H plus or OH minus ions added. Now, if you're adding a strong acid or strong base, it's quite simple to calculate the number of, strong acids, strong bases. They fully dissociate, so it's quite easy to calculate the number of moles of H plus or OH minus ions added. It would just be N equals to CV. But with citric acid, we're going to have to take into account essentially an effective concentration based on the speciation diagram you can see presented for citric acid. So this speciation diagram is based purely on the pKa's, pKb's for a titrant. So the method is completely general, and we can work and titrate with any strong or weak strong acid-base combinations. So for example, at high pHs, citric acid will fully deprotonate and release 3 protons into solution, whereas at low pHs and very acidic conditions, it will not release many, if any, protons at all. And I'm happy to go into any more detail about the method too. It's out in Chem Methods. I just wanted to play a little clip of the pH bot in action. So it's been built and mounted on this 3D printer platform. So essentially we had the movement in Cartesian coordinates XYZ for free. Everything that you see here is being programmed in Python using PySerial to communicate to the objects. We had some highly accurate syringe pumps that could dose our acids and bases to the microliter. We're using quite small volumes of acid and base, and we were looking at formulations up to 15 milliliters. We could autonomously titrate 12 formulations at a time. Here you see one iteration of a titration. And this gives you an idea of the sort of viscosity of the formulations that we had. With the method that we introduced, we were able to reach the set point pH within an average of 3 iterations if we didn't overshoot the set point, or even when we overshot, so we added acid and we need to say add base back, we were still able to recover these samples within a further iteration or two. And you can see we had samples with a broad range of starting pHs, all of which we could titrate to the set point. And we've also released 4 blueprints and for how to build this pH bot. Jumping forward, once I've collected my formulations dataset, I'm now returning to the original problem of the forward model property prediction that I presented on one of the beginning slides. I was able to develop a predictive space stability classifier and also computer vision classifier. And given the reasonably good F1 scores that you see presented, when I've run a SHAP analysis, a Shapley additive explanation, I'm able to now confidently determine and derive some interesting chemical insights. Just in brief, for example, counterintuitively, adding more thickener was not what was resulting in the most viscous formulations being present, and the thickener was causing in fact, a lot of instability. It was, in fact, these self-thickening surfactant systems that were resulting in some rather interesting formulations. We also see particular functional groups contributing to more stable products. One of the other objectives was the turbidity. However, here, unfortunately, as most of the turbid formulations were unstable, the actual turbidity data set that I've collected had a rather skewed distribution, and so when I tried to develop a regressor, this was ultimately quite unsuccessful, unfortunately. But if we're thinking about product design, so initially I've been screening this space, but if we're thinking about product design, then desired shampoo formulation is stable with a low turbidity, high viscosity, and shear thinning properties. And most of the stable samples, they reached the industrial turbidity requirement. Now, probably the most interesting and where there's more data to be utilized. So within the data set, I collected a classification for the zero shear viscosity as very low, low, medium, or high, and also whether the formulations were Newtonian, shear thinning, or another type of non-Newtonian fluid. So this is where I was training a multi-class viscosity classifier. I've also collected the full flow curves in terms of viscosity versus shear rate, so there's more data to be utilized. In terms of this, I was able to train a moderately predictive viscosity classifier, and I tried to improve my model by adding hand-curated molecular descriptors for the surfactants based on our physical chemistry understanding and knowledge of these systems. Unfortunately, we have not managed to, or that degraded the model performance, so I've not yet found the most appropriate set of molecular descriptors, and this is another question for future work. And I hypothesize that instead of these descriptors on individual ingredients, probably we need descriptors, system-level descriptors on the formulation as a whole. So with that, I would like to conclude my talk. I'd like to thank everyone in the SRE group. It's really great this will be I'm moving out of Cambridge today, so it's great to be able to actually spend the day in the conference with a lot of group mates. I'd like to thank the HIP Lab, Kedar, and the group in Singapore. I'd like to thank BASF for co-funding the PhD along with NRF, and Alexei, Kedar, and Dave were the professors I had the privilege of working with. So thank you. And happy to take any questions.